How's everybody doing today?
Pretty good. Hanging in there.
Getting there, hanging in there, hopefully. All right. I've gotten quite a few emails about the paper, which is fine. So I haven't been able to respond to all the emails. Like Sam, I know for sure you wrote yesterday, so I'll get back to you. So don't worry if you haven't gotten the primary research articles you need to finish parts 4 and 5 yet. I'll work with each of you and just kind of see where we're at and make sure you're getting the right papers and all that. So we'll get that sorted out. All right, it looks like it's around 11. I know a lot of people end up joining just like a minute or two after, but um, I wanted to first, oh yes, so first share like inspirational quote, right? Queen Elizabeth was last time. Um, I don't have a really like specific one, but I like this one. It's classic, right? In the midst of chaos, there's also opportunity. I think a lot of these days I'm having a hard time seeing the opportunity. I'm focusing more on the chaos. Maybe you guys feel that way too. So it's a good reminder. Try to, you know, remember there may be some hidden opportunities in kind of what we're dealing with right now. Might not be easy to see all the time. But if you get through this and get through all your classes this semester, um, it's an impressive accomplishment. So that's an opportunity. Okay, that's all I got for inspiration. Anybody else have anything inspiring to share? Has anyone heard anything really wonderful? Any good news? Oh, Jamie, you're saying you weren't able to hear sound. Which recorded lecture was that, Jamie? 4-9. Okay, I'll double check that. Um, Thanks for letting me know. I will see what was going on there. I may have to just redo that one. Okay, yeah, if anything like that happens, please let me know. I'm sort of surprised it hasn't happened sooner, all the technology. Um, yeah, okay. Anything anybody wants to share? Anybody have anything interesting or good news in the world? I'll tell you one thing I think is kind of pretty amazing in Mount Pleasant. I don't know how many of you are still in town, probably not too many of you, but there's a couple different restaurants that have taken it upon themselves to basically just like in addition to the food pantry, be giving free lunches to people who need them. So Max and Emily's has been doing this for a couple weeks now. They have this program called People Helping People. So they're handing out free sandwiches. I think it might be every day. And then um, Hunter's Ale House, they're doing some different things like delivering lunches to like the hospital and medical care facility and things like that. So those are some pretty cool things just happening in the community that I think are really nice. All right. Well, not seeing anything else in our chat. I don't have too much to say today. I know because I'm getting more emails, there's a lot of questions and then potentially stress about trying to, move forward with the research paper. Sections 4 and 5 are much harder than sections 1 to 3. So just keep asking questions, right? If you need help, just keep asking questions and because I think I'll be able to help you move forward if you get stuck. So I did change up our kind of a little bit the upcoming assignments and deadlines. So obviously today was the initial day for the draft of 4 and 5, but we're being a little flexible there. And the idea is that the PowerPoint that I have open right now that is supposed to have narration, so I'll work on that, is where you can learn about gene therapy. And I'll fix that later today because you'll want to be able to look at this assignment to think about treatments that you might want to propose, like new ideas. Um, so there's a short assignment about gene therapy that will be due. That's called the 414 assignment. And it goes along with this nice website that I have listed here. There's a nice example of thinking about gene therapy for cystic fibrosis. So just kind of as a model. So hopefully that's helpful as well. And then the second type of kind of a method for treatments is CRISPR. We've talked about CRISPR a little on and off, like just mentioning it during class. So in this PowerPoint, I'm explaining it. There's a couple videos that help explain it. I know, or I think in the lab 546, if those of you are in the lab class, you're learning about CRISPR. So this will hopefully kind of go along well with that. So those are two different types of treatments that you might want to propose for part 6 of your paper, the final part. So that outline, right, and I've kind of highlighted it on the screen, is now due Sunday the 19th instead of Thursday. So I've pushed that back a little bit so that you have enough time to really kind of learn about gene therapy and microarrays. So that's sort of next week's two lectures. The microarray one's not posted yet, but I'll get that up soon. So you'll want to learn about those before you tackle that outline, okay? And there are also some tip sheets that are now posted in the research paper folder for how you would want to think about structuring those kinds of experiments. So if you've finished parts 4 and 5, you can take a look at the outline assignment and then, start brainstorming a little bit. This is another part of the paper where students often want to bounce ideas off of me. So again, as you're thinking about those, feel free to email with questions. Okay. Okay, so that's kind of just like kind of coming attractions, like what's coming up. Questions about what you're working on right now or about where we're at? Any questions at all? You can type them or you can unmute yourself. All right, while you think about whether you have questions, I'm going to go ahead and show you one more. I'm just going to show you where I've posted these. So let me go into Blackboard. And you guys can see this okay? That I'm in Blackboard? Okay, great. All right, so over here in research paper, I just want to show you where two things are. So one is this finding info for each part that I've referred to a few times. I've been saying it's in the research paper overview folder. So that's right here at the top. It's the third file. And we've looked at that before, but I'll just open it to show you right, that if you scroll through this one, oh, if we scroll to parts 4 and 5, it's got a lot of hints on where to find information within OMIM. All right, so hopefully you've already looked at this, but if you haven't known where it is, or if you haven't opened this file yet and you're still working on sections 4 and 5, I highly, highly recommend you take a look at this document just to help you kind of navigate sections 4 and 5. Okay, so that's right here at the top. And then this new resource here, it's the third kind of folder down. It's tips for part 6, proposing experiments. So if we open the how to design a gene therapy experiment, it kind of just leads you through kind of the logic and thinking about, how you want, does a gene therapy experiment make sense? First of all, that's the first series of questions. And then kind of what kind of information you need to include, because this is always a question. Students are like, okay, I don't know how much detail to put there. So you'll want to make sure you're indicating what model system. And for part 6, when you're proposing experiments, you can't propose experiments in humans. Obviously, we eventually would want to be able to apply the gene therapy to humans, right? To try to help treat and hopefully maybe cure the disease in some cases. But the experimental part right, is going to be a model system. And most often it's going to be the mouse. Then you want to think about the genotype of the model system animal. So if you're trying to cure or treat an animal, you don't want to start with wild type. You actually want to start with a model system, like maybe a mouse knockout, for example. There are a lot of knockout mice that mimic the human diseases. So that would be the starting point for the kind of animal you would want to use. And then you want to think about how you're going to get a normal copy of the gene into the animal. And this is where the 4-9 PowerPoint, the one for today, walks you through these different viral vectors. And then for gene therapy, that same PowerPoint explains the difference between in vivo and ex vivo. And that short assignment also helps you understand that difference. And if you're proposing an in vivo gene therapy experiment, you want to think about how you're going to actually deliver the vector into the animal. Or if it's ex vivo, and if you're doing it in cells, like in cell culture, are you delivering those cells back into the animal? Like for cystic fibrosis in the 4-9 PowerPoint, it talks about how because you want to treat the lungs, you can actually propose using an inhaler, and people have tried this in some clinical trials, right? So cystic fibrosis patients can use an inhaler, and that hopefully can help deliver the virus that has been engineered to contain the wild type CFTR gene and deliver that wild type CFTR gene to the lungs. So that makes sense, an inhaler for any kind of lung disease. But depending on what kind of disease and what tissues are involved, you'll want to think about that. And of course, you want to think about controls. And then the last part is thinking about how you're going to know if your experiment worked. So you want to think about what you're going to assay or measure and how you're going to compare your experimental to your controls and how you're going to interpret that. And in the outline, it walks you through that part as well. Okay, so I just wanted to make sure you know about these tips. There's one as well for microarrays, and I'm going to be adding one for CRISPR. So those are the 3 new methods that in particular for treatments, but also for some other types of experiments, these are some methods that you're going to find, I think, really helpful and interesting to learn about, right? It's kind of like the culmination of a lot of the things we've been doing this semester. And after getting through the nitty gritty of those mapping and cloning experiments, I think this is kind of the exciting part, right? Where we're applying everything that people have learned and being able to apply it and see if we can actually improve treatments or improve diagnosis or better understand, the gene function. Okay, so that's where that is. That's all in the research paper folder in Blackboard. Any questions from anything I just kind of went through? I realize you haven't started thinking about part 6 yet, but if you have finished 4 and 5, I would urge you to start thinking about it right away. It's the kind of, this is the part where you get to be creative. You're not telling me about things people have already done. You're sharing your own ideas. So there's supposed to be some original or novel ideas. Any questions or comments on any of that? I am not seeing questions.
Not necessarily on that, but looking forward to next or like the end of this semester, what are we going to do during the final time? Like, will we have a final time where we have to come in and watch presentations, or how is that
Yeah,
gonna work?
that's a good question. So we have 3 students who would be following sort of the graduate student, um, um, you know, like they have like different metrics for 500 level classes. They have to do things a little bit differently or do a little extra. So in this class, that is to do a presentation. We're not going to try to do it live because I just don't think you can see that like really today we have not so many people, right, who are with us live. I think there's 9 students right here today, and that's okay, right? There's a lot of things going on, and it's hard to necessarily always be able to log on at a certain time, or internet's not working or whatever. So my thought, and I'm gonna email those 3 students, is to have them do what I've been doing, which is to do a narrated PowerPoint It's really actually pretty easy to do, except when the sound doesn't work. So I'll tell them to do a test, and I should be listening to my own to make sure they work. But at any rate, they're pretty easy to do in theory, and then they could post those. And then the idea was that there's usually, when we're on campus, undergrads, have to attend that final class to hear the presentations so that they have an audience, and that you guys have to ask at least one question. So I don't know, I haven't figured out the question part. I'm not sure if there's a way we can do that that would sort of be reasonable. Not sure. So I'll let you guys know, like for the undergrads, which is most of you, right? What your role might be, but there's not a final exam per se, right? So it's certainly not going to be any difficult or right, not a big assessment. The big thing is our paper, right? Does that help, Emily and everybody?
Thank you.
Sure. Yeah. Any other questions or thoughts? All right. Well, I know I have a bunch of emails I need to respond to. I will check on the sound for the 4.9 PowerPoint because that's kind of urgent because I'm telling you to hurry up and get started thinking about that as soon as you finish parts 4 and 5. So it'll be helpful to hear the narration. So I'll get on that right away today, and then I'll just keep helping you guys one-on-one because I am getting quite a few emails, which is good. That's a good thing. Just keep me updated if you run into issues with deadlines, right? And I'll work with you, and we'll make things work for you as well. Okay, so I'll just flip back to this. So just again, just reiterating kind of what we're looking at for next week, a short gene therapy assignment, and then a short microarray assignment. And I'm trying to keep them really short, but I really want you guys to kind of interact with the PowerPoints and make sure you're getting kind of like the main points. So I think by just answering a few questions, it's actually helpful. Hopefully, it's not just, more work in the sense, but I did notice, like, for example, there was no assignment attached to my 331 PowerPoint. And I think that one people didn't really look at or listen to as much as the other ones. And there was some good information in there. So I just want to kind of make sure that I'm encouraging you sufficiently to listen to those. And then once we get to this outline, I think we're really just going to be focusing on making those revisions, and there won't be, I'm not sure if there'll be any more short assignments after the 416, right? So that you can really be focusing on your paper. And, right at that point, you're going to be making revisions from sections 1 to 3 and making revisions from 4 and 5 once I give those back to you. And then we'll be doing the peer editing on April 23rd, which is 1 week before the final papers due. All right. Any last questions or comments from anyone?
I actually wanted to follow up what I said in my email, but I don't want to take up anyone else's time if
Sure, you can just
else
hang
have
on
any.
till people hang up, Sam, if you want.
Okay.
Anybody else have anything? And I'm always happy to, like WebEx one-on-one if you ever want to, but just let me know by email. All right. Well, I'm not seeing any other questions. So if you guys are good, you can log off, and good luck continuing to work on your papers, and email as needed, okay? And hang in there. All right, Sam, looks like it's just you and me.
So not even 20 minutes before class started, I actually found some articles that I was having